Alright everybody, welcome back to Lab 6 of PLPA 303. We are halfway done through this course, so thank you all for sticking around. No assignments due this week or next week, I believe. So just make sure you're keeping your observations if you're here in lab, so that you can turn everything in when it's due in a few weeks. And then for those of you that are taking the course online, just continue to answer everything and turn everything in when it's due. No feedback this week since most of your assignments should be graded in this class. Alright, so today we're going to be continuing on with learning about fungi. We're also going to finish up Koch's postulates, the last step, and we'll be getting into PCR. So that'll be fun. So first things first, let's learn about the fungal groups. The fungi are classified into lower and higher fungi. What we mean by lower is how differentiated they are on the taxonomic tree. So if you guys remember in taxonomy, you'll have branches, and depending on how early on a species branches off, we'll call it either more primitive or lower, and the higher it branches off, we'll call it higher, or more, just higher, I guess would be the term to use. The fungi that we're going to be working with, or learning about today, are two lower fungi, the oomycetes and the zygomycetes. And the higher fungi are going to be the ascomycetes and the basidiomycetes. So starting off with the oomycetes, the first thing I want to say is they're technically not considered to be true fungi. However, they have many fungal-like properties, and that's why we lump them together with them. So the oomycetes are typically found in moist or wet environments, and if you guys recall the hyphae that we were looking at last week, the hyphae of the oomycetes are non-septate or cenocytic, so the nuclei can just move around throughout its hyphae. It also produces two types of spores for reproduction. It produces zoospores and oospores. The zoospores are the asexual spores, so these are just spores that are going to be produced constantly, and they're going to be essentially genetic copies of the oomycete itself. Whereas oospores are going to be the result of fertilization of two sexual spores from two different oomycetes coming together and forming a new, genetically diverse organism. And we call this process oogamy. The female haploid is fertilized by the male oospore. And you can see they have different structures as well. The zoospores are produced from the zoosporangium, and the oospore are produced from, I believe it's the oosporangium. I gotta double check though. But the important part about the oomycetes that you guys need to know is that they're not considered a fungus. Looking at their genomes and looking at and comparing it to other organisms, you'll see that they're actually more similar to algae. Their spores also are motile, meaning they have flagella or flagella-like appendages that allow them to move around, whereas in higher fungi, they lose that ability. So you really only see that in the more primitive fungi. The spores also contain cellulose in the cell walls. So if you recall, in order to consider it a true fungus, you must have the criteria that was established is the cell wall must contain chitin. Whereas the oomycetes don't really have chitin in their cell wall. Some do, but very, very little. It's not a huge proportion of the cell wall. And they have a lot of cellulose as well, whereas the higher fungi do not. So the next lower fungus that we're going to be looking at, or the phylum, is called the zygomycetes. These are the most primitive of the terrestrial fungi. And what we mean by that is the oomycetes are more associated with water and being near water. The zygomycetes are not so reliant on being near a water source. They're also cenocytic in their hyphae, meaning they don't have those discrete cells within their hyphae. And these also produce two different types of spores, and that's, if you guys have noticed in your classes, you probably noticed this in your classes as well, that fungi produce two different types of spores. One type of spore is always going to be the asexual spore, which is just, a genetic clone of itself. And then the sexual spore is going to be, it's going to allow for fertilization, so you get that diversity, genetic diversity. With the zygomycetes, the asexual spore are called sporangiospores, which come out from this sporangium. And the way that these spores are formed is different than in the higher fungi, because whereas with the sporangiums in the zygomycetes, you get this really large structure with a whole bunch of nuclei, and then all the cell walls start to form at the same time. Whereas with the higher fungi, you start to get cells produced like at the tip, and they start to bud off, rather than being produced all at once and then being released one by one. So that's one difference that you'll notice. So we call that cytoplasmic division rather than budding. And then the zygospores are going to be your sexual spores. All right, getting into the higher fungi, the first we're going to talk about is the ascomycetes. And they're called the sac fungi because of the way that their fruiting structures look like. They're the largest phylum of the fungi, meaning they have the most species within it. So if you were to take any random fungus from all the listed fungi, chances are it would be an ascomycete. These fungi do have septate cells, so you will see those discrete cells. Excuse me, septate hyphae. So you will see the discrete cells. And there's also two different stages. What we mean by that is, even though a fungal cell in these higher fungi can have the nucleus from its mother and its father, they don't always come together to form a 2N cell. So what we call those N plus N, if you guys have learned about that in your plant pathology class. So that would be the asexual stage, or the anamorph. And then when those two cells do finally come together, we call it the teleomorph in that sexual stage. And then up here we have the different types of fruiting bodies, or one type of fruiting body from this cup fungus. And you can see the spores form in this structure called an ascus. Plural is asci, or asci. And they bud off rather than form all at once. So the ascospores are the sexual, or the spores that result from sexual reproduction. And as I mentioned, they're contained in an ascus. So this is the reason they're called the sac, because these asci contain many different types of spores within a sac, and they bud off from the top, or whatever the fruiting structure it's within. The different types of fruiting structures will have different names. So cleistothecium, perithecium, and apothecium are some of the types of fruiting structures, and the asci will be contained within the fruiting structures. And depending on the fruiting structures, you can differentiate between the different types of fungi. So these back here are the spores that are produced from sexual reproduction. The asexual reproduction are produced from this, the spores are produced from a structure called a conidia, plural, or conidium, and plural is conidia. And it produces conidia spores. You can see here they kind of look like a long filament, and you can see the individual spores will bud off. They'll just kind of fall off like that, rather than being all formed at once. Alright, so that's it for the ascomycetes. Now for the basidiomycetes. They're similar to the ascomycetes in that they also have a septate hyphae, as well as distinct sexual and asexual forms. So the sporulating structure, which is what produces those sexual spores, is called a basidium. So that's one way you can easily tell with fungi which phylum it belongs into. If you're looking at basidiospores or ascospores, it'll tell you what phylum that fungus belongs to. The basidiospores, or the basidium, I should say, is club-shaped, so it's shaped a lot differently than those seen in the ascomycetes. And the spores are usually in groups of 4. You can see them kind of budding here. It's a little hard to see, but the basidium would be down here, and then the basidiospores would bud off up here. And then for the asexual part, you would also have a conidia, which would be a little different compared to that of the ascomycetes, but they both serve the same purpose in that they produce the asexual spores. So the basidiomycetes contain a lot of plant pathogenic fungi, especially those that cause rust in grass and grassy type of crops. So for example, the rust would be one of the main examples. They're obligate parasites. They can make multiple generations per year, meaning they're constantly producing new spores that can infect new plants. As you probably already know, they infect the stems and then the leaves. Hence why they call it rust, because the Urrutia spores, the asexual spores that it produces, are orange in color. That's what gives it that rust name. They have a complex life cycle as well. If you guys took plant pathology, was it 301? Then you'll know that it alternates hosts. So it's got, I think, 5 different types of spores. And it's categorized based on a microcyclic or macrocyclic lifestyle. That just means how many spores, how many different spore types it has. So macrocyclic is 5 different spore types, and micro is 3, I believe. And then again, whether it's autoecious or heteroecious is how many different types of spores it'll have. Here, I'm not sure what type of spore this is. It's, I'm guessing, based off of the rust over here, that this is some type of Urrutia spore. You can see it's kind of orange in color. And I'm guessing these are the fruiting structures containing those Urrutia spores once it's caused an infection in some plant. And here's some of the fruiting bodies that produce those different types of spores. You've got an aecium, where the spores just kind of fall out. I think this is in the, when we're looking at, what is it, stem rust of wheat, the aeciospores are in the barberry host, if I recall correctly. Then the Urrutia form on the rust, on the stem of the wheat plant. And then these produce Urrutia spores. And then the telium, that is the overwintering spore, if I recall correctly. This will infect its barberry host. Oh, no, excuse me, the spermagonium will infect the barberry host. You guys don't need to know all that. That's just the different types of spores that one single type of fungus can produce. And then there's also the smuts, which, if you guys recall, create these galls that are just completely full of spores and really gross looking on corn. These only produce 1 generation per year though, and they infect the fruit of the plant rather than the whole plant itself. So then any questions then on the fungal part of the lecture? No? Alright, we're going to learn about PCR, which is something that I really like. So PCR stands for polymerase chain reaction, and it's a technique for amplification of a genetic sequence. It was a very significant finding, or technique, when it first came out, because it allowed us to look for specific pieces or specific sequences of DNA. Why, thinking of it from a plant pathology perspective, why would we be interested in finding specific DNA of an organism? Anyone take a guess?
Differentiate.
Yeah, you could use it to differentiate between different types of organisms. But if we want to detect an organism, recall that your DNA of really any organism is unique to that organism. So you won't find, DNA of a cow in a human, because obviously there's no cows in humans. But if, say, with what's going on with the pandemic nowadays, if you find a virus DNA, well, it's an RNA virus, but let's say it was DNA. If you find DNA of a virus in your body, well, then you can say, oh, you're infected, right? Well, we can do the same thing with plants. If we find the DNA of some pathogen in plants, that's because the pathogen is present, even if we can't see the pathogen with the naked eye. So it allows us to see or detect a pathogen even before we're able to see it. And it allows us to detect a specific pathogen as well, not just any pathogen. So the way that works is by amplifying a specific piece of DNA that is unique to that pathogen. So each one of us, even between species, we all have unique pieces of DNA within us. And based on those unique pieces of DNA, we can tell who is who and what is what. The thing is, we have so little DNA within a cell, relatively speaking, of just that one particular type, that it's very difficult to diagnose it, to detect it, I should say. So what this technique does is it not only finds a specific piece of DNA, but it amplifies it, meaning you make a whole bunch of copies of it so that you can detect it easily. Because we're working at the molecular level here, and you really don't need a lot of molecules to properly function, or to have proper function. But in order to detect it, you do need a lot of molecules. So that's what this technique does. So it does those two things. So just to review the DNA structure of DNA, I should say. This is a pretty basic structure, but you can see you've got two different strands. When you twist them out, you've got your forward strand, which is your 5' to 3', if you guys remember that. And then you've got your reverse strand here, which is in red. That'll be your 3' to 5', looking at it from left to right. And it's composed of nucleotides, deoxyribonucleotide triphosphates. In this case, it should be DNMPs. Do you guys remember what is it that holds these nucleotides attached to one another?
Hydrogen bond.
They're all hydrogen bonds, exactly. And how do you know which nucleotides go with what? Like, how do they specifically bind to the other nucleotide? It's just based off of their sequence, right? Your A will pair with your T, and your C's will pair with your G's. So that's all it is. And are hydrogen bonds strong or weak? That's a trick question. They can be strong if you have many of them. However, they're weak in that you can break them just by heating up a sample. Just putting heat into, a little bit of heat into some sample is enough to break apart those hydrogen bonds. And that's part of what this technique does. We break apart those hydrogen bonds. So, it's probably better if I show you guys the video. We're going to watch a video today. everybody see that? All right, so this is also part of what we're going to do today, so you may want to pay attention. So in here, in this tube, we've got our DNA. So we're somehow going to get the DNA of whatever we want to look at, let's say bacteria. We want to figure out what type of bacteria it is. Each bacteria will have its own unique piece of DNA, so depending on what the bacteria is, we'll get different, we'll detect different things. So you got your target DNA, which we can also call our template DNA. That's what they call it in the book. You've got your primers. Primers are just small pieces of DNA that will anneal, meaning they will stick to whatever complementary part of DNA they are, because each primer has its own unique sequence, and it's only going to bind to its complementary sequence. Does everyone understand that? So these we need to design, and you can buy them from manufacturers. So you've got your primers, you got your nucleotides. Nucleotides are just free, you know, A, T, G's, and C's that are just floating around in the solution. And then you've got your polymerase, which is misspelled there, but it's an enzyme that starts to produce an extra piece of DNA when it attaches to a primer. And I'll show you what I mean by that. So all of that gets added into this reaction, and then we'll stick the reaction into the thermal cycler, which we have back there. But let me show you what the reaction actually looks like. So you got your double-stranded DNA from your template, which is your organism, and we're targeting this part of the DNA. We'll say it's some gene that is specific to bacteria. So here they're going to linearize it just to show it easier. Right? All they did was just unwind it. There's 3 parts to one cycle of PCR. The first is the denaturing step. What does it mean to denature something? It means to change its shape or something such that you can't put it back together. So when you denature proteins, you're doing something to them that makes them unable to fold the way they were. Here what it means is you just break apart the DNA double helix, so you have single strands. So if you look closely, you'll see a little gap in between each one of the nucleotides. That's where the hydrogen bond is taking place. So we add heat, which denatures the 2 strands, and you're left with 2 single-stranded pieces of DNA. And then you've also got a polymerase sitting in the solution. So the next comes the annealing step. You got your primers, free nucleotides, yada yada. Okay, so during the annealing step, you see each primer has its own unique sequence, and it's only going to anneal to one side of the DNA. So you got your forward primer here. Oh, this is your forward primer, and this is your reverse primer. In this case, it doesn't really matter, but you can see T is going to pair up with A, T with A, C with G, and the same thing down here. So that's how we're able to target those genes specifically, because the primers are specific. Next, the third step is the extension step, where the polymerase is going to come along and it's going to extend it. So it's going to recognize that primer, it's going to kind of wrap around it, and it's going to use the nucleotides that are floating around in the solution, and it's going to start to add them to the rest of the DNA. See, it's extending it. It's grabbing all these free nucleotides and putting them into the new strand that it's synthesizing. And the result is 2 strands of DNA from 1. You guys see that? So that's only 1 cycle. Now we're going to keep repeating the same process. So if we repeat it, we denature it, we add more primers, the polymerase will come along and use the rest of the free nucleotides to extend it. And now we have 4. And so we're just doubling it from 1 strand of DNA every single time. So that after 30 cycles, you start to get a whole bunch of just this green targeted region. You see that? You do get a couple of these long ones, just because of the nature of it. However, the target DNA is going to be multiplied at a much higher, or much faster rate than the template. We call this the variable length DNA. So you see, at 4 cycles, you've already got equal amounts of template and the target that we're after. and we normally do something like 30. So as the cycles progress, over here it's just keeping count of how many copies you have. So after 13 cycles, you know, we're already in the tens of thousands, hundreds of thousands. And eventually, after 30 cycles, we can have up to 1 billion copies of the same DNA, of the target. And so we have all this DNA sitting in the tube. And now we're able to see if the DNA, the template, coming from the organism that we're interested in, is present, then the primers that we use should anneal and we should get a product from the PCR, right? However, what would happen if we used DNA from some other organism, like E. coli, which we're not targeting? The primers would not be able to anneal to the DNA, and so you wouldn't get any product at all. It would just stay like that. The DNA would just be, it wouldn't form anything. You wouldn't get a product, is what we call it. And so that's how we're able to use PCR to specifically detect a DNA within any sample. So again, you've got your genetic template, which is your DNA. You've got your primers, which are specific to whatever you want to target. And you've got your DNA polymerase, which is going to synthesize that new strand. It also has to be heat resistant, because we have to heat up our DNA to 95 degrees Celsius in order to break that apart. And a lot of proteins are going to break apart at that, or denature at that temperature. So we use the polymerase that was taken from a bacteria which normally lives in hot springs where it's like, you know, almost 100 degrees Celsius. So that's why we use the polymerase from that bacteria. And then you've also got your deoxyribonucleotide triphosphates, which are just your free-floating nucleotides that are going to get incorporated into the product. And this is just a schematic for you guys if you want to see it at home. And again, recall that this is just with, you know, we got almost 1 billion copies just from one single molecule, but in reality we're adding, you know, several hundreds or thousands of molecules when we first start off. So take 1 billion and multiply by however many copies you start off with, and you can see quickly how many times you amplify the DNA. Alright, so that's what we're going to be doing today. We're going to be setting up the reaction for that. But before that, Koch's postulates, step 4. What is step 4? Reisolate was step 3.
What was that, reinoculation?
No, reinoculation, sorry. So we reisolated last week. What is the second part? I get my mistake. What is the second part of step 4 of Koch's postulates? We isolated, but now we need to compare. We need to compare the bacteria to what we isolated during step 2. So what we're going to be doing is we're going to be doing gram staining today for the bacteria that you all streaked out last week, which you isolated from your potato culture. And we're also going to streak it on McConkey's agar. So you're going to be doing those two things. And then we're going to be setting up a PCR reaction as well. So we're going to be doing what's called colony PCR, in which we literally use, rather than extracting DNA from bacteria to use it as a template, we're just going to stick the bacteria, dunk it into the reaction, and then when we heat up the reaction during the PCR, during that initial denaturing phase, it's actually going to break apart the cell, release the DNA into the solution, and then if the primers are able to anneal to that template that we have, then we should get some product in the end. And next week we'll go over how we detect it specifically, because even after we finish up PCR, the tube, you know, the solution that we have just looks the same. So it's not until we run it on an agarose gel that we see something. But we'll go over that next week. So what we're going to be doing is we're going to be creating a master mix, which is just a way of putting all the components that we need into one tube so that we can aliquot it separately. And then after that, we will put DNA of our organisms into each specific tube so that we can test all of them. And this is what your master mix should look like for one reaction. All right, any questions about that? So for you guys online, I'm not going to show you guys how to do the, the gram staining, because we already did that. If you want to see it again, that's in lab 3. And as well as McConkey's agar, it's just regular streaking. Instead, we'll just show you how to create a master mix and then aliquot it. Any questions then before we start? Cool.